Okay. Well, it's not coming. There it is. Okay. They're switching to Windows 11. Has anybody gotten that yet? How much hassle is that? No, the practicum is on the 16th. I put it
Yeah,
on the
okay.
very last day. It's the lecture test that's on the 9th. Okay. And these three tests, and that's all we have left are three tests. These are on the at the very end of the course, and they're really scrunched together. That doesn't make them harder, believe me. It actually makes studying easier because you're studying for all of the exams at the same time, because the material overlaps considerably.
Charlie,
Yep.
is the December 16th our final date of class then?
Yes, it is.
Okay.
Okay. I want to click this off, but I don't want to lose you. So I guess this is the one. Yeah. Let's just go back to modules. I've had classes go until December 22nd before. That's horrible. Horrible. No time to get ready for the holidays if you do get ready for the holidays. Okay, so chapter 16 is the assignment, and we'll take a look at it after the break. When we come back, I'll cut that chapter down a bit. Our lecture topic today is the autonomic nervous system. It's the start of it, actually. And if we bump ahead to the 18th, it's the autonomic nervous system combined with the spinal nerves. There are actually three lectures on the nervous system going backwards. This one on the 18th, this one on the 16th, and we already had the first one. It was way back here on 1028. So we have quite a bit of separation between the two. I don't think that's going to be a problem. If you find it's a problem uh, and you can't review and patch it together, then we can do that together. Okay. I'm not going to give you this lecture again right now. I will if I have to. This was the first one. I just want to remind you, that's all. We talked about the nervous system, its functions, the components, the central and peripheral nervous system, and then the organization of the nervous system, central part here, all this, the peripheral portion, the incoming or afferent fibers, the outgoing or efferent fibers. Okay. Then we talked about the glial cells, which make up 90% of the central nervous system mass. And the first type of glial cell was the astrocyte, which holds things together. This is the astrocyte here. Okay, going real fast, but we've had it. Here's the oligodendrocyte, so I think this is blue or blue-green, and they attach around the axon of a neuron to help speed transmission. Okay. Astrocytes are also present at the same time, but it's the oligodendrocyte that sheaths the axon and makes the myelin sheath. The microglia is the cleanup cell. Types of neurons, we had the multipolar neuron, the most common type of neuron, and we took this one apart. We use this diagram here to label or identify the parts of the neuron. The bipolar neuron, one way in, one way out, one dendrite, one axon. doesn't allow for any changes. It perceives one thing, such as a wavelength of light, and not the other wavelengths of light in the eye uh, to propagate that wavelength if it's present to the brain. The pseudo unipolar neuron, that's usually found in the integumentary system in the skin, and we have incoming and outgoing. It actually has two axons, the incoming and the outgoing. Okay, they're really essentially the same thing, mirror image of each other. But these perceptive areas here are called dendrites, and they receive pressure, pain, heat, cold. Okay. Send the information to the cell body, which is located off center, and then forward to the axon branches. I know this says axon terminal, but I'll remind you that it's properly called an axon branch or an teleodendrion as well. All those terms apply. And then I gave you some vocabulary that we went through. Okay, this is sort of an introduction then to the autonomic nervous system. And if we jump down to this little chart, we can see it mimics the chart that we saw just a moment ago. Okay. Here's the central nervous system, however. Only difference on this chart is it's not located at the top, it's located here. And it communicates with the peripheral nervous system. Peripheral nervous system receives information or stimuli from the body, okay, and the organs. The somatic sensory system, that would be pressure, vibration, touch, pain, come in and they make their way to the peripheral nervous system and into the central nervous system. Since they're incoming, they're called afferent. Okay, so these neurons and the nerves that they eventually compose are afferent. This is also known as the afferent nervous system. Leaving with motor commands is the efferent nervous system or the motor division. And this takes motor information to the voluntary nervous system, here called the somatic nervous system. This one innervates skeletal muscle. So this is sort of just typical. We're not getting into anything autonomic yet. Okay. To get into autonomic, we have the outgoing or motor muscle. Excuse me. We have motor impulses going to the autonomic nervous system, of which you're not supposed to know anything yet. And don't worry if you don't. When I was a kid in junior high school, I did a project on the autonomic nervous system, and nobody could figure it out, even my teacher at the time. So they figured if I could figure it out, that they let me win the science fair, and I did with nothing but charts. I don't know that I really understand it myself even today, but I love thinking about it, and I hope you enjoy it too. Okay, the autonomic nervous system is split into two parts, the parasympathetic and the sympathetic. Okay, we'll go into that in just a moment. Okay, backing up from the chart, let's see what we skipped. Okay. The afferent nervous system is fairly easy to understand. The efferent nervous system is also fairly easy to understand when you're looking at it on paper. Okay. In the afferent nervous system, incoming, the sensory stimuli are initiated by receptor cells and they move to the CNS. We talked about this once before. In moving from the environment to the CNS, a stimulus only has to cross two neurons. Now, there may actually be, in some instances, more than two neurons in the pathway. Okay. But I was talking about the least number, mainly to shock people that the two neurons is all that you actually need, and two neurons. So I'll use the example of an elephant stepping on a thorn. Well, that must be painful, okay? And that pain reaches the central nervous system by way of two afferent neurons, incoming neurons. If you only have two neurons in a pathway and one of them is damaged or killed, then the pathway will not work. And if that's the only two neurons that are in that pathway, you're going to lose function. Well, we don't just have two neurons. We have sets of two neurons. We have many, many redundancies so that instead of just having two neurons in one pathway, we have thousands running side by side. And we call that structure that they form a nerve. Okay, so nerves much bigger than a single neuron. Okay. There's safety in numbers, meaning that if one of them gets damaged, diseased, or whatever, you got the rest of them to take charge. In the efferent nervous system, that's the outgoing or motor nervous system, the situation changes. Okay. The outgoing impulses are channeled to the voluntary or the involuntary nervous system. Okay, so here we're using better vocabulary, in my opinion, than the chart does. This is the voluntary, and this is the involuntary. Motor commands that reach the voluntary nervous system innervate skeletal muscle. The word innervate, people eventually think that they know what it means, and they usually do. It's one of those things that you can catch on to. But let's get it up front here. When something is innervated, it is acted upon. That's all there is to it. It's acted upon. So in the voluntary nervous system, skeletal muscle is innervated. In the involuntary nervous system, the innervation is of smooth muscle, cardiac muscle, and the glandular epithelium. All things that we are told, and we can argue a little bit with, we're told that we don't have control over. Well, we do have a little bit of control over these things, but not nearly as much as we do in the voluntary nervous system and the skeletal muscle. Okay, you need to remember autonomic nervous system controls or innervates smooth muscle, cardiac muscle, and the glands. Okay. When we talked about the skeletal muscle, like the biceps and triceps, always a good example. They're antagonistic to each other in those cases. All skeletal muscles, except for the facial muscles, have an antagonistic counterpart. So that if you flex your arm with your biceps, your triceps can extend your arm and get it back to a previous condition. You have muscles working against muscles. Okay, all of course work to keep you going. Well, we have two divisions in the autonomic nervous system: the sympathetic and the parasympathetic. The sympathetic division literally jumps into action when there's danger, when we're in a fight or flight situation. We're going to run away, or we're going to stay and confront it. Okay. Now, we don't have to have a menacing person or animal attacking us for the sympathetic division to work. There are other things that can cause it to work too, and sometimes abnormally. People who have occasional, I hope only occasional for them, panic attacks. You have a panic attack and you feel it, you know it. I hear so many people say, "Oh God, that's just so hard. I had a panic attack." They didn't really. You know, if you really know, and I know many of you do, what a panic attack is, uh, it builds upon itself. You react to it, and unfortunately, it's a sympathetic division that is a little bit out of control here. Okay? I used to have panic attacks, and I mean real ones. And the first time I had one, I was 4 o'clock in the morning at my house in Majeska, putting a lecture together on all things houseplants, a group of plants that I didn't even know. I was up late, I was up early, and now I'm driving into school the 25 miles, and I'm the first one there about 6 in the morning, 7 in the morning. I'm down in my office on the first floor of the science math building, and there's nobody around. And I get my notes that I had made for this lecture that I had to give in an hour or two. And I I still do take lectures seriously. I had written out notes, and damn, guess what? I left them at home. I didn't have the notes that I needed. And this was not an easy subject. It was all Latin names and such. I grabbed books off the shelf and I just threw them around the room. I just threw them at the wall. I got so mad at myself. I'm the only one there, so I figure that's okay. But I initiated my first panic attack, and it got worse and worse, and I couldn't understand it. I didn't know what was going on. Went outside. There were a few cars. I went over and kicked their tires, trying to get rid of this panic attack. And remember, this one instructor did show up. I felt like I was going to get sick, and I went into the restroom, and I'm hanging on to the basin, water basin or whatever, just trying to stand up. And this guy comes in, he says, "How you doing?" You know, like people do. And I said, "Not so good." Well, I guess he hadn't expected that answer, you know. So he said, "Well, hang in there." Oh Christ! I could have killed him. I could have killed him right then and there if I felt I could have moved. Anyway, by this time the secretaries had come in, and one of them recognized that I was in some distress. I was just going crazy, and she drove me over to the hospital. We went inside, and this second I got in the door in the emergency room, they said, "Have you ever had a panic attack before?" I mean, they'd seen it, and that went on for quite some time until I guess I got over it. Let's see, I can't see that. Okay, so when the sympathetic division goes a little out of whack here, okay, uh, it can lead to one of those. The parasympathetic division is a less stressful environment. You have to do things like digest food. You have to have the stomach's smooth muscle operating. Okay, you have to have the small and large intestine operating, and you have mostly your control through the parasympathetic system. The sympathetic system also innervates the same organs. Okay, all organs have a sympathetic and a parasympathetic attachment. For the stomach, the sympathetic division, in a period of time of fight or flight, the sympathetic division would tell the stomach, stop. We don't need any digestion, just stop. We need the energy and everything elsewhere. Okay. So these two can work opposite each other. They're always working. Both of them work all of the time. If you can recall the chart without now having it in front of you, the autonomic nervous system, our abbreviation, is a subdivision of the peripheral nervous system, and it innervates the smooth cardiac muscles and the glandular epithelium. The ANS is controlled by the hypothalamus, which we learned in P3. The two opposing fractions are the sympathetic and the parasympathetic, which we just talked about. There's a little warning here that if you were reading this only, okay, you might find it a little difficult the first time through. Use your book, reread, or look again at chapter 12 as you go along with this, and it should fall into place. You can always email me if there's a difficulty. We haven't run into anything I could consider difficult yet, though. Again, this is text repeating what I just said, okay? 2 neurons is the pathway in the autonomic nervous system to reach the central nervous system. And also coming from the central nervous system and going to the peripheral nervous system, the motor pathway to the organ being innervated, whether it's skeletal muscle or smooth muscle, cardiac muscle, or glandular epithelium, is also only 2 neurons long. Let me say that again. Coming in, okay, stepping on the thorn again, 2 neurons to reach the central nervous system. That's just to reach it. From there, you might send the information to the brain, and the brain can think about it and use up hundreds or thousands of neurons, but it only took 2 to reach the central nervous system. If the central nervous system decides to react, it'll send out a motor impulse. Sometimes it will decide that no action is needed. Okay? The brain will think and decide no action may be needed. If it is needed, a motor impulse goes out, okay? And there will be a 2-neuron pathway if we're going to the autonomic nervous system to reach the innervated items. The innervated items, again, being skeletal muscle, smooth muscle, cardiac, and glandular epithelium. There are 2 neurons reaching out to the organs. The first is called a preganglionic neuron. This carries the impulse to the second neuron, which is the postganglionic neuron, which should have been in bold-faced. I always mean to go back and change these, but unless I write them down, I sometimes forget too. It says the same thing though. Preganglionic neuron, postganglionic neuron. Here they are in the picture down here. This one following the arrow. This is the preganglionic neuron. It's being called the presynaptic cell here. That's correct. But a cell of the nervous system is a neuron. And this is preganglionic, but they're calling it presynaptic. Well, the junction between any 2 neurons is a synapse. So it's okay to call this presynaptic and this one the postsynaptic neuron. But we just said we don't find just 2 neurons in a pathway. We have a cable of neurons forming a large cluster, which we call a nerve. Okay. And when synapses come together, not just this one, but hundreds or thousands of them in a nerve, they all gather at the same spot. And that is a ganglion. A synapse is one crossing. A ganglion is lots of crossings at the same point. So that's where we get the term that I used here, preganglionic neuron. And then after you cross an individual synapse of the ganglion, the postganglionic neuron. This postganglionic neuron runs the distance to the thing that's being innervated. It's either going to go to, again, skeletal muscle, smooth muscle, cardiac muscle, or glandular epithelium. There's a name for the junction at the end here. We'll get to it later, but I want to just name it now so that when we get there, it's sort of reinforced. The junction between the postganglionic neuron and the organ, say the stomach, say the smooth muscle of the stomach, is a myoneurojunction. A myoneurojunction, muscle nerve junction. It can also be called a neuromuscular junction just by reversing the order, but it means exactly the same thing. Action potential. Action potential refers to the impulse generated by the axon hillock, which would be right here, and traveling down the axon. Okay. This is a good representation of a synapse. On the left, we have an axon branch, a teleodendrion. Okay. It's also known as an axon terminal. The other side is the dendrite of the next neuron, and there is a space in between. The space, which we'll see later in a good micrograph, the space is the synaptic cleft or division. When an impulse comes down the axon here and terminates in the axon branch here, it will cause the release of neurotransmitter. These little dots here, and here they are here, and prior to that, they're located in these synaptic vesicles. This neurotransmitter has to literally swim across or move across by diffusion, across the synaptic cleft until it reaches and stimulates the dendrite. So here's some that have gotten across. If only a few get across, only one dendrite might get stimulated. That may not be enough to propagate the impulse. We need more. They're all headed towards the axon hillock. The depolarization is headed towards the axon hillock. And if we don't get enough depolarization of the axon hillock, then there'll be no further movement of the impulse down the next axon. Don't worry in this picture about enzyme, just neurotransmitter. There are two main neurotransmitters, and we'll talk about those in just a second. Don't worry about mitochondria. All cells, and these are cells, have mitochondria. Synaptic vesicle empties neurotransmitter into the synaptic cleft. The entire arrangement here, axon branch, dendrite, and cleft, that comprises the synapse. Okay, here's a section of the spinal cord, which at first would seem out of place, but it's not. Okay. Over on this side, on the left, red indicates part of the afferent nervous system. It comes in, coming in through a spinal nerve, and then, since the incoming from the skin especially is by pseudo-unipolar neurons, here's the cell body off center. Remember, we saw it being an incoming axon, an outgoing axon. Okay, and here's the cytoplasm and cell body, also known as a parakaryon. The impulse continues in and enters into the dorsal horn. Okay. Now this only shows one neuron. There's going to be thousands of neurons coming in. And not just from your little finger, but all your fingers, palm of your hand, the back of your hand, your elbow, all parts of the integumentary system. Well, this is literally going to be jammed, and the cell bodies are going to be jamming up here. And that's why this is swollen, enlarged. And what is this? It's called the dorsal root ganglion. All impulses coming in enter through the dorsal side of the vertebra and then into the spinal column. They'll enter through the dorsal horn right here. They can now go up or down. Usually they'll go up to the brain and come back down, and then they can leave. Sometimes it's just a quick trip. Look, come in, there's an interneuron, goes across, and goodbye. You're going to a motor function. That's called the reflex arc, a reflex arc. If you've ever done something foolish, or sometimes it's not foolish, it's just it wasn't expected. But a foolish thing is, oh, you got an electric stove, and you don't remember which one of the burners is hot, and you, for some reason, want to know which burner is hot, so you touch the burners. Oh, you find the hot one, all right, and your hand jerks back without you thinking about it. That's a reflex arc. And then you jerk back even before the brain has been told how stupid you were. And then the brain will take over and it may tell other muscles to get the guy the hell over to the sink and get some cold water on it real quick. Okay. A reflex arc doesn't go up to the brain, it just comes right straight back. Most things will go to the brain eventually, but the reflex arc takes care of itself. The sympathetic nervous system is also known as the thoracolumbar because the preganglionic neurons arise in and emerge from the thoracic and lumbar vertebra only. Well, that's so much wordage there. We have to remind ourselves that in the autonomic nervous system, it's a two-neuron system, preganglionic and postganglionic. The preganglionic neuron is the first one, and it has to have communication with the central nervous system. The central nervous system is telling the preganglionic neuron to send this impulse and get it started. And in the sympathetic nervous system, all of these impulses for the sympathetic nervous system arise from nerves, spinal nerves, coming out of the thoracic and lumbar vertebra only. In the parasympathetic system, the preganglionic neurons originate in the brain and in the sacrum. So it's called the cranial sacral system. Parasympathetic nervous system is also the cranial sacral system. Here's a comparison between the two divisions. The sympathetic system is known as the thoracolumbar system. We just said that. Nerves originate from the spinal cord in the thoracic and lumbar regions. In the parasympathetic system, it's known as the craniosacral system because nerves originate from the brain and the sacrum. The sympathetic system generally excites and prepares us for fight or flight. Both systems, however, are active at all times. The parasympathetic system dominates in less tense situations, like digestion. Both systems still are active at all times. This I'll tell you now, and we'll see it in a moment. In the sympathetic system, that's this one on the left, the preganglionic neuron, remember there's a pre and a post. The preganglionic neuron is short, relatively short, and it terminates in something called the chain ganglia. We know the chain ganglia from P3, I believe, and we also there called it sympathetic trunk ganglia. Okay, we'll see it again in just a moment. The postganglionic neuron is comparatively long, so it's short and long, whereas anatomically in the parasympathetic system, the preganglionic neuron is long and may be so long it terminates right next to the organ, then a short little postganglionic neuron is present. And so the second one is short. Why this is important? Haven't a clue. Does it have to be that way? I don't think so, but I'm not the authority, and I haven't seen the authority say why it has to be that way. But it is that way, and it's a good way for us to identify what we're looking at. Sympathetic system, its two neurons are short and long. In the parasympathetic system, they're long and short. The sympathetic system is adrenergic. I'm going to say this without explaining it. I got a picture to show it to you in just a second. At the synapse between the preganglionic neuron and the postganglionic neuron, the neurotransmitter is acetylcholine. Let's jump over here. In the parasympathetic system, the neurotransmitter between the pre and post ganglionic neuron is also acetylcholine. Both systems start with acetylcholine as the neurotransmitter between the pre and post ganglionic neurons. But when the postganglionic neuron gets to what it's going to innervate, okay, gets to the myo-neuro junction or the neuromuscular junction. In the sympathetic system, it gets a boost of epinephrine. Boost of epinephrine and fires it up essentially, gets it going faster. Whereas the myo-neuro junction in the parasympathetic has the chemical as acetylcholine again. The parasympathetic system has acetylcholine at both junctions, the one between the pre and post ganglionic neurons and at the myo-neuro junction. Because it's going to be acetylcholine both times, the parasympathetic system is described as cholinergic, whereas the sympathetic system has adrenaline or norepinephrine or noradrenaline. I don't care what you want to call it. Different forms of this are used by this system. Okay. But since then, the neuromuscular junction uses the PEPI chemical here. It's called adrenergic, despite the fact that it also uses acetylcholine. in the first synapse. Now, I hope the rest of this is not new. It's meant to describe, get you more familiar with this. There's nothing new here. Okay, here's a lot of the organs of the body, and you'll see they're duplicated, like the lungs on both sides, the heart on both sides. That's because the sympathetic and parasympathetic divisions all go to all organs. Okay. We'll start with the sympathetic division, the fight or flight division. It starts from here. This is to represent the first thoracic vertebra level, second, third, fourth. I wonder if they got it actually right. It'll be down to the fifth lumbar level. And the nerve comes out. I'm going to pick this one. The nerve comes out, and here's where it can synapse. This is the sympathetic chain ganglion, also known as the trunk ganglion, sympathetic trunk ganglion. What you see on this side is a chain. That's why they call it chain ganglia. It's about 1.5 inches to 2 inches away from the center line of your back. And the same chain would be on this side too. Everything would, of course, be bilateral or duplicated. So here's a short neuron, and then a longer one, this one going to the eye. Here's a short neuron, and a longer second or post-ganglionic neuron going to the salivary glands, going to the lung, a short neuron, and then a long neuron. When we go the other side, okay, coming from the brain here, vagus nerve comes down to the lungs, and that's the first neuron. And right there, there's a little tiny spot. That's the second neuron, very small. Same thing down here. Let's go to the heart, a long neuron, and then a short neuron, and then the myoneural junction. So it's short and long for the sympathetic, long and short for the parasympathetic. And some also do come from the sacrum, like the ones that control the bladder. Here's our first, long, and then short. Now, the interesting thing here, and I'm going to make a big deal of it on Thursday, is the elevator action. I can pick any one of these. Let's do the lungs. In the sympathetic system, the preganglionic neuron looks like it's synapsing right here. Okay, it's synapsing in a ganglion. These are known as the chain ganglia or sympathetic trunk ganglia. But it doesn't have to synapse there. It can actually now go right up the elevator or right down the elevator. Theoretically, it could go all the way up and down. And if you stimulate all of these at one time in a fit of fight or flight, they can all reach here. They can all go their own way, up and down. Remember, one nerve is many, many neurons, and it can split and go upwards and downwards. Those pathways pre-exist. Those pathways don't develop just because there's a stimulus. The pathway is there waiting to be used. So you can actually stimulate the entire damn body by getting to the chain ganglia from just one or two places, and then send impulses up and down and then out. I have an exercise for you on Thursday that will really make that sink in. Don't worry if it didn't sink in at all just yet. Yeah. This little sketch is to show a sympathetic pathway versus a parasympathetic pathway. Two neurons, and then the organ that's innervated. In the sympathetic chain, you have a short preganglionic neuron. And then you have a neurotransmitter, acetylcholine, that goes across, stimulates the postganglionic neuron. And then you have a neurotransmitter at the neuromyonuclear junction, and it's noradrenaline. That's what the NA stands for in the diagram. It doesn't stand for not applicable. Okay. So this is the speedy one. In the parasympathetic system, you start with a preganglionic neuron that is long. The neurotransmitter is acetylcholine. A short postganglionic neuron, and the neurotransmitter again is acetylcholine. Okay. Every time you have a synapse to cross, and it doesn't matter which system you're in, you're going to slow down the nerve impulse. Well, why would you want to slow it down? Wouldn't necessarily. But every time you have a synapse, you probably in the peripheral nervous system have many, many synapses, a ganglion instead, so that you can actually switch tracks, exchange information, and get the impulse going, not just to one single cell someplace, but to an entire organ. These are the cranial nerves, and you will be responsible for knowing these. The most important part of knowing them is just to be able to identify them by their name and number. The number is always given in Roman numerals. It's not acceptable to use Arabic numbers or anything else when you're writing. Okay, when you're talking, yeah, we don't have words for these. We just use 1, 2, 3, 4, 5, up through 12. If I ask what is cranial nerve number 2, the answer would be optic. What does it do? What's its sensory function? Okay, you need to know its function. Some have 2 functions, like this one, number 7. Some have only 1 function. Some have a sensory function, but no motor function. So for instance, obviously, the olfactory nerve would be for smell, but it won't initiate anything, not itself. Maybe the smell would alarm us if it smelled like smoke, and we'd do something. But that nerve is only for smell. The optic nerve is only for vision. Okay. Down here, numbers 9 and 10, the vagus nerve especially, have both sensory and motor functions. As simply as they're written here is all you have to do. Remember them as. So practice that And it will be on lecture test 3. Lecture test 3. Okay, let's 3:05. Let's take a 10-minute break. 3:15. Okay, we're back. Okay, can you hear me?
Yes.
Thank you. I've decided not to go through chapter 16 page by page with you, but instead, I'll send you an email like I always do to everybody, outlining the parts that are important. Okay. It just looks like it's going to take quite a bit of time that well, I'd rather spend it elsewhere. So I'll get it to you by Thursday. Any questions? Okay. Okay. You're seeing week 13?
Yes.
Okay, thank you. And this time I want to do that. I've shown you this one before, but it really pertains to this last section that we're in, the fourth unit. Okay, you've seen this one, but just bear with it for a moment. I put this handout here, and you've seen it. It names the glands, it names the organs that you're going to be responsible for. In what way? You need to be able to identify them. You need to be able to give their function if they have a function. Almost everything does have a function, but there are things, that don't have a function. Things do not have to have a function just because they exist. For instance, uh, earlobes, do they have a function for humans? I mean, yes, we have made a function for them, place to hang things or clip things on, okay? But we don't necessarily have to look for a function for everything. What I've done here, uh, in some cases, I have given a function, but more importantly, I've put down what type of tissue uh, they're composed of. Okay? And that's the material in blue. Okay. Lots of stuff, but a lot of this we've gone over. This we haven't. Integumentary system we have. Endocrine system, no. The kidney we've gone over. Okay, so a lot of material is double dipping. Okay. Organs and systems, part 1, and tissues, part 1. Want to try to get through both of them. The first one should be the easier. because we've gone over a lot of these structures before. Yes, and when you open it, Sasha, it does say 12.1 up there, and that's just something to be ignored.
Okay, I printed
Okay,
it
and
anyway.
it says 12.1 down here too. I fix a lot of these, but you know, I kick myself for ever putting the dates in there. You know, I thought originally this lockdown pandemic thing was only going to last half a semester.
Me too.
Yeah, so
Thank you.
now I've got to go back and try to fix them. It's not necessary if you know what's going on. Okay. Okay. Well, this one's not going to be that bad. I've shown you some of these slides before. Okay. I'm going to go through it as if I have not shown you anything, because that would be the like review then. And this one, this is a neuron in the deep blue or purple. Okay. And the longest appendage coming off would be the axon, no matter which way it's forming or flowing. Here would be the perikaryon, and here would be approximately the axon hillock. Dendrites are out here. We don't get to see the other end. They're very fragile and didn't persist in this slide. These little blue dots are the nuclei of the glial cells. That'd be the astrocytes, oligodendrocytes, and the microglia. And of course, that's lecture material that we've had already, and it's good to review. Anytime I say something that seems like it should be reviewed to you, but you don't quite remember it, go ahead and ask. Okay. This structure down here is the salivary gland. I always say with the salivary gland, a lot of people don't recognize it right away. And when you get to a slide you don't recognize, have in mind the name of a slide that you always have trouble with. That's going to be the one. This is a salivary gland. Okay. Now, it has lobes and divisions between the lobes. That means it's therefore a gland. Okay. If it's an exocrine gland, one that secretes fluids by way of ducts, well, then there has to be ducts. There's a duct, a duct, a duct, small ducts right here. These cells here in the exocrine gland secrete enzymes, the darker cells. Those are called acinar cells. ACINAR. Here it is down here. There it is. Acinar cells secrete enzymes. Even in the pancreas, we have acinar cells, but we also have cells that secrete hormones in the pancreas. None of that goes on here. This salivary gland is a merocrine gland, meaning it secretes fluids only. And since it is a gland, it's composed of glandular epithelium. We don't distinguish epithelial cells, glandular epithelial cells, in the salivary glands versus the pancreas versus the liver. Here's the tongue. At the top of the tongue is stratified squamous epithelium. And lining these passageways downward are the taste buds. Taste buds are what really give the tongue away. When I put it on a test, I notice in face-to-face classes where I can see all the people that as soon as that one comes up on the screen, they just immediately write it down. Okay, I know I showed you this, but like I said, it's bears reviewing. This happens to be esophagus. I wouldn't expect you to know that right off the bat. You might be able to figure it out though. You're looking at an epithelium because it's lining. You've got a lumen here. An epithelium always covers their lines. And cells on the outside are mostly flat, not all, but mostly. And it's multi-layered. That means it's stratified, squamous epithelium. Now we have stratified squamous epithelium on the surface of our skin, on our epidermis, but this isn't the surface of the skin because it's around a lumen. So this is inside. The only place inside you're going to find it, upper esophagus. This is the stomach. Okay. Here's the lumen of the stomach. It's a fuller. That's the lumen up there. And the wall of the stomach looks smooth. This is an artifact of the preparation. If I were going to show you this one on a test, I would not have the artifact in there. I'd just chop it here. Okay. Down below, we have smooth muscle. We have circular smooth muscle and longitudinal smooth muscle. Not knowing exactly which way the slide was cut, I'd be happy if you just recognized that that was smooth muscle. Okay. These are called, this one especially, since it's exaggeratedly large, a gastric pit, a gastric pit, gastric pit. They go downward. And the enzymes for digestion and hydrochloric acid go into the stomach once they're produced. This is the colon, or large intestine. Now, normally we would jump to small intestine. I don't know why we didn't, but that's fine. It's a beautiful picture. On the colon, we have a lot of light colored cells called goblet cells. As we move through the GI tract, you'll find a few of the goblet cells in the stomach, more in the small intestine, but tons of them here, like suckers on an octopus in the colon itself. These are not villi because these are not projections up. This is not a projection up. This is a separation downward called an intestinal crypt. Just like in the stomach. Stomach has no villi. The colon has no villi. Its function is reabsorption of water, compaction of waste. This is the pancreas. This is a combined type gland. First off, here is an islet of Langerhans. Here's an islet of Langerhans. Here's another one right here. This is a slice between, this is connective tissue holding lobes together. Whenever you find a long slice like this, you have a gland. There's a gland there, and that's connective tissue. There's also a duct down here, and the pancreas is allowed to have ducts to get rid of pancreatic amylase produced by the acinar cells, which are the cells we didn't point out a moment ago. We pointed out instead the islet of Langerhans. The islet of Langerhans has two types of cells, alpha and beta. Alpha cells produce glucagon. Beta cells produce the insulin, and those are taken up by the blood and distributed to every part of the body. Whether any part of the body needs it or not, it's going to get it. A hormone goes everywhere equally, but destinations don't necessarily react to a hormone the same way, and some ignore it completely. Look at this line here. This isn't two pictures. This is the connective tissue connecting this lobe and this lobe.
Charlie,
Yes,
what are those white spots?
say it again.
What are those white spots?
Here,
Big ones, yes.
these are ducts.
Okay, thank you.
This one looks like a duct. You can see the cells are on the outside. This one sort of does, but it's cut more obliquely. Okay, on the liver, one of my favorite slides actually, you have little tiles. They're actually 6-sided tiles, but this picture doesn't do a good job of showing that. It also looks like the underside of a sand dollar. We used to have a lot of sand dollars around here. When's the last time you saw one? Probably years and years ago in our area. Okay, and then in the center of the tile, if we call it that, which is really a lobule, is the central vein. Central vein. There's some smaller veins too, but one central vein. Blood is delivered to the liver at the triad, where 1, 2, 3 lobes come together. It leaves a triangular area over here too. Okay, and you get a small artery bringing blood to the liver. You get a hepatic portal vein coming from the small intestine, bringing suspect blood to the liver. And you have also a bile duct. We'll see those on models and it'll be quite clear. The liver has to have blood of its own, and it's got to be clean blood that the liver has actually previously decontaminated, if that's the best word, or detoxified. So that's brought by a small artery. But the contaminated or suspected blood that just absorbed by the small intestine comes by way of the hepatic portal vein and comes to the liver. That blood spills out all over these cells, all through them, 3-dimensional now, and they all head towards the central vein. And as they go past these cells, they are detoxified. So alcohol, for instance, a certain amount of is detoxified with every passage. Certainly, the liver isn't able to detoxify 100% of a toxin out of one passage. If it were, nobody who liked to drink beer or anything like that is going to bother because they'll never feel it. It would be useless. Okay. Unfortunately, we have some things that we can ingest that are so poisonous that they kill us on the first pass. Mushrooms, for instance, certain mushrooms. And there's no evolved mechanism to change that because they kill you. You're out of the gene pool immediately. And so there'll be no evolution to change something that toxic. Potatoes are toxic. Oh, you're thinking French fries. Yeah, sure. And all that grease and things. I'm thinking of just raw potatoes, have solanine in them. A little bit isn't going to hurt you. But if you hang potatoes in a basket in the kitchen, like some people do, they sometimes turn green. Don't eat a green potato. That's the presence of solanine. It can easily make you sick. Okay, I showed you this one with the eye. Here's the, start with the dark part, choroid coat, and then decide which is retina and which is sclera. Well, this is retina over here with the multi-layers, and here's the sclera. Here's the lung. Okay, and what I don't know what else you could mistake it for. The type of tissue that we want to bring out is the thinnest right here. Thinnest right here. Some of it's not as thin and some has 2 layers, but where it is just one thin layer going across, that is simple squamous epithelium. Simple squamous epithelium is found in the lungs then. It's not the only thing in the lungs, but it's there. And we also find simple squamous in Bowman's capsule. Here's the skin, okay, and the dark part all the way down to here is epidermis. Dermis is underneath. The first layer of the epidermis is the stratum basal, and it's only 1 cell layer thick. The next layer would be the stratum spinosum. We would know that just because it is the next layer. But after that, we're going to be guessing, unless we have some chemical to dye elidin, hyalokeratin, and keratin. But we know from the text of the integumentary system lecture that we have 5 layers. We have the stratum basal, stratum spinosum, granulosum, lucidum, and then this one would be the stratum corneum. Yeah. This is in the dermis, and the things that are to me obvious, maybe it isn't, of course, to the person who's seeing it for the first time, but the sebaceous gland or oil gland is here. That's a gland, so it's got to be lobed. Here's another lobe of it. These are connected. Okay. Here's another one. Here's another one. Okay. Sebaceous glands secrete sebum. They're holocrine glands because they secrete entire cells and not just fluid. There's some adipose scattered in here, a light area. This set of tubules that happens to look like a dancing dog, this is a pseudoriferous gland, a sweat gland, and that's of course a American gland that it secretes only fluids. Oh, since this is all dermis here, it's a mixture of stuff. Okay. We refer to this as dense, irregular connective tissue. Dense, irregular connective tissue. What's it connecting? It's connecting the upper skin, the epidermis, with the muscle below. Here's an ovary. The ovary is a sex organ, but it is also an endocrine gland. It secretes progesterone from this structure. This is an egg or ovum located inside of the follicle. Here's one too, but it hasn't developed. In getting stimulus in the pituitary gland, this one didn't react. This one did, to form a mature follicle referred to as a Graafian follicle. This is a very helpful model. We start here with follicles, and once its follicle stimulating hormone from the pituitary reaches the ovary, it will stimulate the development of a follicle and the egg inside. Here's the erupted follicle. The egg presumably has been expelled and hopefully reaches a good destination. Okay. Now, the erupted follicle begins to change into this structure. This is the corpus lucidum. The corpus lucidum secretes progesterone, which tells the pituitary gland to quit making follicle stimulating hormone. We don't need it right now. We're waiting to see if pregnancy has occurred. Okay? And if it does occur, then the corpus luteum continues to make more progesterone to turn off the follicle stimulating hormone. If a pregnancy doesn't occur or is terminated, then the corpus luteum will quit acting to produce progesterone. And then it will deteriorate to either here or here to a structure called the corpus albicans. The corpus albicans has no further function. It will stay in the ovary forever and do nothing more. Here are seminiferous tubules. Okay. The seminiferous tubules here located in the testes. Testes, of course, are sex organs and produce spermatids, but they're also an endocrine gland. They produce testosterone. Okay. Okay. That took us away from this one. We finished with that for today. And I want to do the tissue. Get us moving. Okay, there are 4 tissue types. We know that. We've done it already, but bears repeating. Epithelial tissue, connective tissue. We have muscle tissue and nervous tissue. And the first one and only one we'll do today is the epithelial tissue. Okay. There are different types of epithelial tissues, and they're all categorized by their shape or the arrangement of their cells. By the arrangement, I really mean the numbers. Is it a single layer, a double layer, a triple layer, or more? That's how we categorize epithelial tissue. The basic shapes are cuboid. Now these cells down here are cuboid. Okay. Look like a dice. This looks like a whole bunch of dinner rolls or dinner rolls on some server. Columnar, cuboid and columnar, and flat for squamous. Flat as a pancake. The arrangements are simple, meaning a single layer. Compound if it's 2 layers thick, and stratified if it's more layers thick. This first one is simple because it's only 1 layer. That layer is attached to a basement membrane. Follow it here, this colored portion. Connective tissue is underneath that, which would go to the muscular area. Okay. But the cells are cuboid, and the cells are in a single layer. So this is simple cuboidal epithelium. This wouldn't do for your skin because it wouldn't be thick enough to protect you. But we do use it elsewhere. Okay, now imagine you take this layer, flexible pan, and let's roll it up. Take this end and roll it here. Take this end, roll it here. And you roll it together like this or like this. Where is the basement membrane in this picture? Is it inside or outside?
Outside?
Outside. It's outside. Okay. The lumen itself is lined by the cells here. Okay. And this one, we've made a tubule. And this tubule is simple cuboidal. Now, this diagram over here definitely shows simple and cuboidal with the nucleus in the center. Okay, that's what we like to see under the microscope. But sometimes we don't get that. And I didn't even think of it, but the book that I took this from, look at this. These look more columnar, don't they? Yeah. Well, they're actually simple cuboidal. How do you know? Because you look at all around on the slide, and you find other ones, and they do look cuboidal. Simple cuboidal. The most prominent place to find simple cuboidal cells is in the kidney. Okay. In the kidney tubules out here. Now, this is a glomerulus. Then we have a space, small space, glomerular space. And that's surrounded by Bowman's capsule. And I have better pictures of that for you, but we've already actually talked about it. So, but these tubules over here are lined by simple cuboidal cells. These are the kidney tubules. They almost look like necklaces or bracelets. That one's particularly good. Here's another. Simple cuboidal. Now, where it looks like it's 2 layers thick, it is not. This is another tube touching the back of this tube. This tube is touching that tube. And of course, that would make it feel and look like 2 layers thick. But this is cuboidal epithelium. And it would be located in the tubules of the kidney. Simple squamous is our second type of epithelium. The first type is simple cuboidal. Now we'll do simple squamous. And that's in Bowman's capsule. Here's the glomerulus. Here's the glomerular space. And remember, I talked about a soccer ball. You know, if you put your fist inside of it, your hand or fist would be the glomerulus, but you'd be inside of the ball, the skin of the ball. And that's just, and it's very thin. It's Bowman's capsule. And it's simple squamous. Where else do we find simple squamous? We did it 10 minutes ago. The lung. The third type of epithelial tissue is simple columnar. Now, normally people like to see this picture turned 180 degrees, so the fingers are up. It doesn't matter. My God. Okay, so this is a villus in the small intestine. A villus in the small intestine. These are the microvilli, or the epithelial layer. Inside, this is inside is the lamina propria. The muscularis mucosa is not showing, the third layer, but these are villi. Projections inward that greatly increase the surface area for absorption and digestion. Look at the epithelial layer. It's single, it's simple, and it's also columnar. These are definitely columnar. This is really nice. We're not going to cover compound cuboidal and compound columnar. Why do I bother even writing it here? Because I gave you a chart of pictures, and these are listed there in parentheses. I showed it to you. They're in parentheses because we're not going to cover these. I wrote it here as well, just to keep everything on track. Stratified squamous. We've already had our lecture on the integumentary system. So here we can review it really quick. The outermost layer is the stratum corneum, which protects us from desiccation. Under that is the stratum lucidum, which might increase in size if we have a lot of activity, physical activity. The stratum granulosum, spinosum, and stratum basal. There's 5 distinct layers, but how many layers of cells? My God, some of these have 4, 40 different layers. We have many multiple layers. So this is stratified. It's not 1, it's not 2 layers thick, it's multiple layers thick. This is stratified in the final layer, first being formed down here, then pushed up. The final layer is flattened. So this is stratified squamous. And we said a moment ago, stratified squamous would be the outer skin, the epidermis, or we could also find it on the upper esophagus. We also said we could find stratified squamous one other place. Anybody happen to remember? The tongue. The tongue. Transitional epithelium. The name implies that it can change its shape. It can morph. Okay. This is a multi-layered epithelium, the transitional epithelium. Okay. And it does stretch when the contents that it surrounds increases. It will stretch. And then it can regain its shape when the contents are emptied. We especially think of the ureter when it's filled with urine. The ureter stretches and the inside cells against the lumen will also flatten or stretch, but regain their shape when the urine is released. Here's what it looks like. The lumen is out here. The transitional epithelium. You might think, oh, that's a villus. No, it's not. Look how many layers it's got. 1, 2, 3, 4. This is multi-layered. 5, 6 over here. Okay. A villus has only a single layer of epithelial cells. And they're all stratified. They're all simple columnar. Okay. This is a relaxed projection here, though. These cells aren't flat. It's only when they stretch, they will stretch flat. Okay. And then when the urine is released, they regain their shape again. When you see multiple layers like this, it can only be one of 2 things. Stratified squamous, okay, in which case the outer layer would always be flattened, or transitional. And so this one has to be transitional. When we make a slide from an animal, my guess is the animal is dead and in a very relaxed state at that point. So we're always going to find the transitional here in a relaxed state, where the outer layers of the multi-layer event here are relaxed, chubby. This one can be very pretty under the microscope when you get used to finding it. This is pseudostratified ciliated columnar epithelium. Hell of a name, but every part of that gives it away as to what it is. Start with the second word, ciliated. See the hairs? Those cilia beat and they move dust in particles of any type in one direction, usually out of the body. We find this in the lungs and in the nasal cavity. We find it in the respiratory system in general. This is in the respiratory system to knock the material out. Where does it go? Where does this material go? If it's coming up from your chest, you swallow it. Okay, I guess if you got a cold or whatever, you can spit it out. But when you don't know that this is going on all of the time with you, you end up swallowing it, whatever it is. No harm done. Good job done, actually. Okay, columnar refers to the column-like shape, certainly not squatty. Okay. Now, some are squattier than others, like this one. But we always look around for the majority. Epithelium, coats or lines, and that means that coats or lines, in this case, the lungs or nasal cavity. And pseudostratified, that's the key word. When you look at this, how many layers thick is it? Is it just one layer thick? Or maybe 2 layers thick? Or 3 layers thick? You can't really tell. Well, the fact is, you begin to learn this one just by seeing it, and you see that silly, dead giveaway. This is 1 layer thick. The basement membrane is down here, and every one of these cells is attached to the basement membrane. In other words, there's only 1 layer. Why does it look like there's more than 1 layer? Because some cells are small, others are taller, and then they arch over other cells, and it makes it look like you've got 2 or even 3, because you're not seeing the entire cell. It's hidden behind the first one. Imagine you're way back in kindergarten, 1st grade, and the school hires a photographer to take your picture. I've had it taken many times. Class of 1954. Oh, jeez. And well, all the kids lined up, and the photographer realized, of course, there are too many in width, too many kids. So let's double up, okay? Instead, double up. And there were still too many kids. So we tripled up. And the kids on the front sat down on the ground. The kids in the middle leaned over their heads, leaned down over the heads of the first group. And the kids in the back could stand upright. Okay, so I've got 3 rows. And the photographer is going to see all the heads of the 3 rows of kids. But every kid is standing or sitting on the ground on the basement membrane. Okay. They're just obscuring portions of each other. So when we see this under the microscope, and we've gotten used to looking for the cilia, we know that this is pseudo-stratified, ciliated columnar epithelium. There's a little chart at the end of this, if you care to fill it out. I'm not collecting it, okay? The name of the tissue, text page where you might want to locate it, come back to it later, function and location. Well, you can also get that from the chart I was referring to. You can use the histology study guide and be filling this out as you go through, because this one is due by P4. Okay. along with the reading assignments you'll have by then. I'll get you 16 as I promised. And histology slides. Okay, these are the slides we just did. We did not do the 2 in parentheses, as I said. We call the stratified cuboidal and stratified columnar. We will be doing all the rest of them. Okay. And you can work ahead if you want to. We don't have to. Okay. I have a lot of stuff coming between now and next month when we finish up, but we don't want to burn out at this point. People are doing really well, and so hang in there. Make use of me if you have any questions, and that's that. Are there any questions?
I have a question. At
Yes.
this point, typically, do the students maintain their grades or does it dip?
You don't want to do not count on maintaining your grade. Okay, because there are so many of you whose grade average, right? I don't grade on averages. When I look at your average, that's meaningless, except to give me a clue, and you too. Your grade is based on points. So you add your points together, and you want to get 720 for an A, 640 for a B. Okay, but it's a quick thing to do to see what your average is and where you're going. And there are a lot of people in here sitting on 90 and 89. Now, how can I tell which way they're going to go? I'm not going to. We've had a lot of extra credit, and I hope they'll maintain or do better. Now, a lot of people, they've got their grade locked in already. They know it. They're not going to have any trouble. Okay, and others can improve, but the ones I worry about as an instructor are the ones that are right there on the borderline. Okay, because they're going to expect me to find a way to give them points at the end, and I'm not going to do that. I'm not going to do that. You got to earn it. Okay, so you can. I'm not worried about that. If you will, that's what I worry about. Okay, I answered the question, maybe not the way you wanted to hear, but
Thank you.
okay. Okay, then if you want to hang around, you can, and if you want to go do something else, I'll see you on Thursday, and we'll tackle the spinal nerves. Don't miss Thursday.
Thank
Okay,
you.
bye-bye.
Thank you.
Thank
Bye.
you, Marley.
Bye.
Thank you, Charlie.
Thank you.
Bye.
Thank you.
Bye, Mike.
Bye, Charlie.